So now let's move to South America. We have an incredible researcher who is a senior researcher of the National Science Foundation of Argentina. He has a PhD in biochemistry from Argentina. He is a professor at the Universidad Nacional de La Plata in south of Buenos Aires. He is an expert on longevity, and he's an expert on cryonics. I don't know if he will talk about that or if he can talk about that, but he is the leader of cryonics in South America. So welcome to Rodolfo.
Okay, so I will speak not about cryonics, but about rejuvenation by cell reprogramming. So in this brief presentation, what I want to do is to share with you a look at an emerging avenue in life extension, namely rejuvenation by cell reprogramming. Let's begin with a reflection on rejuvenation. The longing of man for immortality and eternal youth is universal and time immemorial. It is in our nature. We will always seek to defeat death, or at least to postpone it indefinitely. In the Middle Ages, alchemists, a blend of mystics and proto-chemists, tried to synthesize a mysterious potion, the elixir of eternal youth, able to confer eternal youth to those that dare to drink it. Now, it seems that science has found the fountain of biological rejuvenation, the cytoplasm of the oocyte. So it seems that at long last, we are beginning to undertake the conquest of biological rejuvenation and perhaps immortality through science and technology, the hallmarks of our species. The story of biological rejuvenation began over 5 decades ago with the discovery of animal cloning by John Gurdon and collaborators in the UK, in England. So this slide summarizes one of the typical experiments they did at the time. So what they did was to collect intestine cells from frogs, cultured them, and then isolated their nuclei. In parallel, they collected oocytes from frogs and fertilized oocytes. They inactivated their nuclei with UV radiation, and then they transfer these somatic nuclei from the intestinal cells into the enucleated oocyte. Shortly after that, the cytoplasm of the oocyte reprogrammed those somatic nuclei, which become embryonic nuclei, and start setting in motion the program of developmental program for a new frog, which in the end gave rise to a full, healthy adult frog. So this seminal study demonstrated that the fate of somatic cells is not fixed. Animal cloning was achieved 30 years later with the birth of Dolly, the sheep, and shortly after that, other mammalian species were cloned. So animal cloning finally, that was in 1996, got the credentials and the credibility of a truly feasible technology. And there was a very transcendental implication in the discovery of animal cloning. So this implication was that in the cytoplasm of the oocyte, of the mature oocyte, there are molecules able to reprogram a somatic nucleus by activating the developmental program of the individual. What it remained unknown in 1996 was how complex was the constellations of factors necessary to reprogram a somatic nucleus. But this enigma was elucidated only 10 years later with the discovery of cell reprogramming. In effect, in 2006, Takahashi and Yamanaka in Japan published a paper, a seminal paper really, showing that the transfer of 4 master genes, KLF4, SOC2, OCT4, and CMYC, to mouse fibroblasts in culture was able, after a couple of weeks of action, to reprogram those cells into cells that behaved as embryonic stem cells. They called these cells induced pluripotent stem cells, or iPS cells, and the four genes that they used are now known as the Yamanaka genes. So the discovery of cell reprogramming made it possible to implement rejuvenation, cell rejuvenation, in cells from old animals and humans. There is a core of mostly recent studies, less than 10, independent studies, confirming that cell rejuvenation can be achieved by cell reprogramming. Because of time, I will only describe two of these important studies. This study was published in 2011 by a French group led by Jean-Marc Lemaitre in Montpellier. And so they used skin cells from healthy centenarians. They cultured those cells, and as expected, they confirmed that these cells showed abnormalities, not because that the holder was a diseased person, but because they were old. So they studied a set of parameters and found, as expected, that there was a typical transcriptional signature of old cells in these cells. There was shortened telomeres, as expected, dysfunctional mitochondria, high levels of oxidative stress, and reduced proliferation potential. This is in contrast what one would observe in a skin cell from a young person, which of course will be quite normal. So using a cocktail of 6 reprogramming genes, including the 4 Yamanaka genes, they reprogrammed those cells into iPS cells, which of course were quite young, then incubated those cells with a cocktail of factors known as differentiation factors that induce the cells to become again a skin cell. When they studied the same parameters in these cells, newly generated cells, they found that they showed a typical transcriptional signature of young cells. They had elongated telomeres back to youthful lengths. Their mitochondria were back to functional levels. They had low levels of oxidative stress, and they have recovered a youthful proliferation potential. So, according to these parameters, the skin cell from a young individual and these cells were really indistinguishable. So they concluded that this cell had been rejuvenated. In December 2016, another groundbreaking paper was published by the group led by Juan Carlos Ispizua Belmonte at the Salk Institute in La Jolla, California. They used transgenic mice, and these animals carried a sequence of DNA. I represent one of the cells of the animals here for the sake of simplicity, of clarity. So each of the cells of these animals carried a sequence of 4 genes, the 4 Yamanaka genes, under the control of a regulatory sequence, or known also as a promoter. So under basal conditions, the Yamanaka genes were silent. But when they added the antibiotic doxycycline to the cells, it bound to a regulatory protein. This protein bound to the promoter, to this regulatory promoter, and the 4 genes were turned on. But when they removed the antibiotic, the system became inactive again, and the genes were silent. So transgenic progenic mice are animals that have a really fast aging process. They age very prematurely, and by 5 months of age, they are already old. So Belmonte's group used 5-month-old transgenic progenic mice, and they added to the drinking water the antibiotic doxycycline, and so that turned on the 4 Yamanaka genes. Cell reprogramming began in each of the cells of the animals, but 2 days later, they remove the antibiotic, therefore interrupting the reprogramming process. They let the animals to rest 5 days, sorry, and then added doxycycline again for 2 days. Reprogramming began, removed the antibiotic 2 days later, resting out the animal for 5 days, doxycycline again, and so on. So what they did was really what is called cyclic partial reprogramming. And after a number of cycles of partial reprogramming, they sacrificed some of the treated animals, compared them with some untreated controls, and studied a number of organs, viscera, stomach, spleen, kidney, and skin. They studied the organs anatomically and histologically, and found out that in the treated animals, some of the signs, anatomical and histological signs of aging, had been either attenuated or eliminated altogether. Some other signs of aging were not affected by the treatment. Furthermore, the treated animals lived 50% longer than the non-transgenic progenic mice. So it was really a quite significant life extension. Well, this was the first implementation of tissue rejuvenation in vivo. And of course, as a first study, it had limitations. But it was a proof of concept study anyway. The two more relevant limitations were that when they interrupted the reprogramming process, the cyclic reprogramming process, the signs of aging came back. So they were not permanent rejuvenation changes. And the second shortcoming of the study is that we couldn't use transgenic humans for rejuvenation. So these are two limitations, significant limitations that the study had. But I think that in the not too distant future, these hurdles will be overcome, and mature rejuvenation technologies may allow humans, non-transgenic humans, of course, to be rejuvenated, and the rejuvenation changes to be permanent. We have constructed a system similar but not identical to the system that used DNA system Ispizua Belmonte. Our system also has the 4 Yamanaka genes, but also expresses the gene for green fluorescent protein, a reporter gene. In other aspects, it's the same as the Yamanaka system, the Ispizua Belmonte system. Then, when we provide to the animals doxycycline to the drinking water, and the 4 Yamanaka genes are expressed, the cells of the animal will become fluorescent, which is a quite convenient feature. Our animals will not be progenic animals, will be normal animals, that is wild-type animals, because we want to study natural aging, not pathologic aging. Once we have our animals, next year, hopefully, we will follow Belmonte's steps and try to implement in vivo rejuvenation. If we succeed, we will focus our analysis on the function and structure of brain and female reproductive system. Well, at this point, it's worth mentioning that now a quite accurate biomarker of age is available. It is known as the epigenetic clock. It is based on the extent of methylation of 353 methylation sites on human DNA. Just let's say a word about DNA methylation. Methyl molecules are very small molecules. They are attached to very specific sites of DNA on other molecules. They usually play a regulatory role. In DNA, they usually act as gene expression repressors. So we are born with most of our 353 methylation sites on DNA methylated. But as time goes by, there is a progressive demethylation on those sites. So in 2013, Steve Horvath at UCLA devised a mathematical algorithm known as the epigenetic clock that uses the percentage of methylation of each of these 353 sites. And when you feed the algorithm with those data, it will provide an age in years for humans, in weeks for mice. And it has been shown that what is known as epigenetic age or DNA methylation age, has a very high correlation with the chronological age, in this case, of mice. This is really a quite reliable system. And using this algorithm, Horvath, in his paper of 2013, collected skin cells from a 30-year-old healthy volunteer, reprogrammed them into iPS cells, measured their epigenetic age, and found out that the epigenetic age was less than 1 year, much less. So the epigenetic clock is a really wonderful tool to determine whether a rejuvenation treatment really works or not. It has demonstrated that cell reprogramming does rejuvenate cells. Well, I want to close this presentation with just mentioning two implications that the epigenetic model of aging has. The first one is represented here. So here we have two cells, a sperm cell and an oocyte. Let's say from a young, these are you humans, from young person, say, let's say a 25-year-old woman and a man. So because 25 years have elapsed since they were born, their epigenetic clock will be a little bit displaced. So when this couple conceives a new individual, the first cell of this new being, the zygote, will have its epigenetic clock reset to 0 by the reprogramming factors present in the cytoplasm oocyte. So the descendants of this couple will not inherit any of the age of their parents. And I believe that the same phenomenon explains why we age as individuals, but life doesn't. DNA has remained as young as always since the emergence of complex animals on Earth, some 550 million years ago, at the time of the Cambrian explosion. So this is a replica of the previous slide. So let's call this couple generation 1. At generation 2, the next couple, when they conceive a baby, the zygote will still again have its epigenetic clock reset to 0. Same will happen to the third generation, and so on. So at generation 1,000 or 10,000, the new individuals, the new babies that are born, will be as young as they were at generation 1. So, in my opinion, the epigenetic model of aging elegantly explains why life doesn't age. Science fiction writer Arthur C. Clarke stated for his third law of science and technology that any sufficiently advanced technology is indistinguishable from magic. It seems now that the magic of human technology is making rejuvenation possible. Thank you.
So let's see the hands up quickly. We need lights, please, lights. Okay, one question there. Who else? Another one there, too. And then 3. Okay, 1, 2, and 3. Sorry, I saw fourth one, but only 3 questions. Okay, let's go quickly.
Hello?
Yeah.
Great, thank you. Thank you so much. That was fantastic, the clarity of Michael's presentation. I feel clarity is so important for action. So thank you. I love the way your brain works. The quantified human, that was fantastic. I think everybody should see that lecture just to see how quantified we can become. I love the idea of life not aging. itself. And I've noticed that over the years as a physician, that life really yearns to live itself. I wanted to ask about the Horvat. Is that going to be a biological marker test that we're going to see coming?
Okay, quick questions first. All the questions first. Second question, please.
Hi, it's just in response to the last presentation about in vivo cell replacement, and I've read a lot about this, but like, is there anything on the horizon where you take a drug that maybe has all the Yamanaka factors and it basically reprograms a small portion of your cells throughout your whole body? Because I'm aware like this discovery was like made 12 years ago, but there doesn't. I haven't heard like about a specific drug or sets of RNA in a delivery capsule that gets round to all the cells in the body. So, and at the end of the day, like that's what you need to do, like to have cellular restoration. So, if there's developments going on that I'm unaware of, I know AgeX are working on some of this, but I don't know the details of it.
Okay, third question, please, quickly. Sorry for the rush, but we have two incredible shows that you will never forget after this.
Okay, sorry for my English. I wanted to know if other epigenetic mechanisms like ubiquitination, phosphorylation, RNA of interference and so have been tested and watched over, and what's their role in this aging process or anti-aging process? Thank you.
Yeah, thank you. So let's go for quick answers, please.
So it has been tested, the Horvath method, in thousands of clinical samples, taking the methylation data from databases, and some correlations, clinical correlations, have been found or established with the dates, the ages of the algorithm, and they are really quite consistent. But this is only for healthy individuals. And a pathological individual will have an epigenetic age higher than a healthy individual. So will not be a 60-year-old person. If it is healthy, will be 60 years old for the epigenetic clock. If it is an Alzheimer patient, it may have 70 years old epigenetic age. So it's quite useful and revealing a biomarker.
Okay, thank you. Other answers, Michael and Ben?
There weren't any questions for me, but I want to make, concerning plans B and C on my diagram, I've been to conferences, aging conferences all over the world, and wrote about them in Life Extension magazine. I have a pile of them here, and I invite people to come up and get a copy.
And Michael?
No, nothing to add, thank you.
Okay, sorry for the rush, but we are going to have plenty of time in the conference and tonight.